sensory input, where's the motor output? Like I said, this is to test, for you to test your own comprehension. So just do the best you can for memory. I'm not going to grade it, I'm not going to judge. And I'll show you the answer eventually. So, did anybody not get a copy of the course syllabus? And it's on Friday. And then Wednesday I handed out this. This is a copy of the Colloquium on Learning Strategies. This is the poster on learning assignment. I handed it out on Wednesday. Anyone not get a copy on Wednesday? There you go. We'll talk about this a little bit more at the end of class. Doesn't have to be pretty. Just as long as you get all the right neurons in the direction of definition. Take about 30 seconds to finish up. Ready? So here's the big view, right? So remember, the reflex arc goes through the spinal cord, doesn't go to the brain, which means it's very fast. You start with the sensory neuron. The sensory neuron picks up a stretch in the muscle. So this is a sensory cell that's within the muscle fiber itself. It's not anything to do with vision or touch or anything like that. There are actually a whole host of receptors inside your body, your muscles and your joints, that give you sensory information. It's actually, a lot of people call it a sixth sense. It's called proprioception. And it's basically the awareness of where all the parts of your body are located in space, even without any visual or other sensory input. So for example, if I stand here and close my eyes, I raise my left arm, I know my left arm is raised, not from any visual cue, but because of proprioception. Because I have receptors in my shoulder and my muscle that tell me that I've just raised my arm. So you have a sensory cell, it goes to the spinal column. There's an interneuron here that it connects with. Then these connect with motor neurons. And one of them innervates the quadriceps muscle, causes it to contract. The other one innervates the hamstring muscle, but this actually doesn't send an impulse, so the hamstring relaxes, and that's how your leg can kick forward. You always have to have one muscle contracting while the opposite muscle relaxes. And the information flow, of course, goes from sensory to inter to motor. Okay? Good. Any questions about that? Reflex arcs, very simple but very useful. Okay, remember we talked about the brain? Okay. Draw me a brain. It's a side view, so it's actually half a brain. That's okay. And take a look at your parts and list as many of their functions as you can remember. Take a few seconds to finish up. Ready? Okay. Here we go. So this is the cerebrum. Now remember, we talked in more detail about the different parts of the cerebrum. But if I asked you to do that, I asked you to draw me the cerebrum. I asked you to draw me a brain, which is this whole thing. So the cerebrum, of course, is the largest part of the brain, and again, we're talking about human beings here. It has the motor and sensory cortex. Remember, it also has the sensory input areas like hearing and sight and smell, and that has integration areas. So it puts together things like hearing and speech. It puts together things like vision and text and reading and comprehension. So that's all in the cerebellum. We call it thinking part of the brain. The diencephalon, remember, it starts out in the embryo very close to the front of the head, but now it's deep inside the brain, deep inside in the middle of the brain. This is where we're going to be today. We're going to talk about the diencephalon because really that's where most of our learning and memory are stored, as well as emotions. So those are part of the diencephalon. The diencephalon is the hypothalamus and the thalamus.
Yeah, just recording the
class. Cerebellum, right, at the back of the brain, that helps to traffic motor and sensory information between the cerebrum and the brainstem. The brainstem has the midbrain pons and medulla. These mediate these signals going to and from the spinal cord. So sensory signals come up here, get relayed through the cerebellum into the cerebrum. Actions, decisions to move your muscles go from the motor cortex through the cerebellum to the hindbrain, down the spinal cord, to the muscles that actually do the work. Also, remember the hindbrain or the brainstem is our sort of our very primitive fish brain. It controls a lot of the very basic functions like respiration, heartbeat, things like that, which is why you can be brain dead where your cerebrum is no longer functioning. But your heart can still beat, you can still breathe because the hindbrain is still alive. So the cerebrum is the most delicate part of the brain, so usually when you get head trauma, that's usually the first part to be affected. So there's your brain. Remember the 4 parts and basically what their functions are. Okay, any questions on that? No? Good. Okay. You can always ask questions. Please don't feel like intimidated to ask a question. There's no such thing as a stupid question. If you ask a stupid question, I'll laugh privately. I won't laugh. Okay, so we're going to talk about learning and memory today. The neural basis of learning and memory, how we learn and remember things and how that information is stored in our brain. So when you learn something, right, it's really part of learning and remembering. So it starts with preparation. Like you're reading your notes, you're doing homework, you're reading a textbook to get background knowledge. Then you want to take in a fact, a piece of information. Say the cerebrum is the thinking part of the brain. That's going to go into your short-term memory. Then when you review this fact later on, it's going to end in a feedback loop. Where the fact returns to your short-term memory again and again. And eventually, when something is in this loop long enough, it's considered by the brain to be important information. This is something that should be stored. And at that point, it enters your long-term memory. And that's when it's basically stored and cataloged and indexed so that you can retrieve it. It's not too different from a computer hard drive, really. And that, when it's in long-term memory, that's what allows you to retrieve that information when you're taking an exam. This is not anybody's brain. Probably my brain right about now, but yeah. So that's generally how learning and memory go. Input through your eyes, through your hands, through your ears, whatever. Feedback loop in your short-term memory, transfer to long-term memory, and then index for retrieval. And that's how you remember something. So the key to learning and remembering things is a phenomenon called neuroplasticity. And basically what it means is that when adult neurons communicate, they can change. So one way adult neurons can change is by the addition or removal of synapses. Right? So here are two neurons, N1 and N2, and they both have an input into this third neuron, just N1. And this looks like a peripheral neuron, but I don't know. But basically, when you are recalling things, when you are learning something, you may see that more input comes from N1 into this cell, and so the number of synapses from N1 to this cell will grow from 1 to 3. But there's not a lot of input coming in from N2, and so eventually N2, this one synapse completely degenerates, disappears, and N2 no longer has a synaptic connection to N1. So by recruiting different or extra synaptic inputs, that's how you create a memory. So really, there are a couple of ways to do it. One is the changing, actually I'm going to put proprioception up here because it's not proprioception. But basically, two ways that your brain remembers things. One is a change in the number of synapses. And the second one, which is pictured down here, is a change in the strength of synapses. So in this case, there are two synapses with this neuron. This one is stronger, this one is weaker. When you learn something, this synapse becomes stronger. What that means is that there's a stronger postsynaptic response. So the depolarization, when this big neuron here receives a stimulus, an input, is much stronger after you learn something. So you can either change the number of synapses, or you can change the strength of synapses, or both. And both of these processes, changing the number of synaptic connections or the strength of synaptic connections, are activity dependent. What that means is these neurons have to be firing action potential in order for these changes to take place. These don't take place in the absence of any sort of neural activity, which is why one of the things about active learning and about review and repetition is that it continually stimulates these neurons. And so it allows them to strengthen the synapse. It allows them to create multiple synaptic inputs where before there was only one. So it's not a passive process, basically. Learning is an active process from the brain all the way out to you and your fingertips. So all these changes have to be the result of continuous repeated brain activity, which is why active learning is such an important part of learning. So plasticity is the key, right? Plasticity, plastic, moldable, changeable. So the term plasticity is used quite commonly with other things, but neuroplasticity basically means nerve cells can change their connections, make them, break them, make them weaker, make them stronger. And that's really what memory is about. It's not about generating new neurons. It's about changing the connections between existing neurons. So, as part of memory, you have two components of your memory. You have short and long-term memory. You also have sensory input. So sensory input brings information in, whether it's through your eyes, you're reading something, your ears, you're hearing something, your hands, you're taking notes. That's sensory input that's going into your short-term memory. The short-term memory is located in the hippocampus. Slide up there. But short-term memory is temporary, it doesn't stay. And so things have to be transferred to long-term memory if you want to remember them. And long-term memory is in the cerebral cortex, because that's where lots of cells and lots of connections are. And so the analogy to a computer is not that different, right? So there is, a computer has inputs just like a human body, right? You get inputs from a disk or from another computer. The computer has short-term memory, right? information in its CPU and the RAM, but that memory is not permanent. If you turn off the power to the computer, that memory is lost. But if the memory is thought to be important enough, if the user is writing a document and you want to save it, right, because it's important, you'll transfer it to long-term memory, which is usually on some kind of hard drive storage, whether it's a regular hard drive, or whether it's a solid state drive, or whether it's a memory stick. Right? That's analogous to long-term memory. So again, similar with humans, right? You can put things in short-term memory for a computer or for a human being, but they can't stay too long. It's not permanent. In order for it to be permanent, it has to go to your hard drive, or in human terms, your cerebral cortex. So you can think a lot about learning and memory as how a computer works. Right? Information gets stored short term, then it gets put in long term. And just like a hard drive, right, there's a catalog. So if you say, look for information on the hippocampus, if you think, what do I remember about the hippocampus? Then your cerebral cortex is going to look through all your long-term memories and say, okay, here are some things that you remember about the hippocampus, because they have the tag hippocampus on it. So that's how you know you remember things. So here is the hippocampus. It's a bilateral structure at the center of the brain. It kind of looks like a C, but remember, your brain has two sides, two hemispheres. So there's one in each, right down in the middle. Experiments have showed that the hippocampus, the short-term memory in humans, can hold between 5 and 9 bits of information at a time. And a bit is defined as a letter or a picture or a word. So a bit of information. And that's stored in the hippocampus. But these bits of information are only stored for about 20 seconds before one of two things happen. Either they're forgotten, and usually what that means is that there's another piece of incoming information that displaces that, right? So you can only hold between 5 and 9 bits of information, which means if you're learning more stuff, some of the stuff you had in your hippocampus gets lost because it only has a limited storage capacity. Or it gets transferred to your long-term memory, which is in your cerebral cortex. It is your hippocampus. Here's connections to the cerebral cortex. So that's why it's important to repeat things, because your short-term memory loses things after a while. So, and it may not be 20 seconds, sometimes it could be longer, sometimes it could be a few minutes. There's no set standard for how long things stay in short-term memory, but it's volatile. They don't stay there for forever. So, if you learn something in class and then you review your notes that night, and then you read it in the textbook the next morning, that's where the things that get put in short-term memory finally gets transferred to long-term memory. So keep hitting that same thing. Hey, this piece of information, whatever is important, put it in long-term memory. Of course, there's not a lot we still don't understand about how that works. We don't understand how things that are retained in long-term memory can be accessed by you using, say, words or concepts or thoughts, right? We don't know how you say, what do I know about the hippocampus? And all these memories come up. We're not really sure how that happens. But there's a lot of really interesting stuff going on right now. And I remember I talked about fMRI, functional magnetic resonance imaging, where you can actually image working neurons in the brain. And you can actually image people, the brains of people asked to recall pieces of information that they learned. And you can look at all the places in the brain that have activity. You can try to conduct or formulate a hypothesis that might explain how that works. But we're still really in the infancy of that. There's a lot that we don't know about the brain. We know a lot about neurons. We know a lot about neural cells. We know a lot about behaviors that people have, and the things that they learn. But in the middle, how we get from those nerve connections to people remembering things like your mom's face or your grandmother's cookies is not very clear. So lots of interesting stuff still to be learned about how we learn how we remember things. So one of the consequences of this increase in the strength of synapses is a phenomenon called long-term potentiation, LTP. So when the strength of a synapse is increased, and that's called long-term potentiation. That strengthens the synapse. So interestingly, the strengthening of this synapse doesn't occur in the presynaptic cell. It occurs in the postsynaptic cell. So generally, the neurotransmitter that's used in learning and memory is called glutamate. And there are two receptors for glutamate in the central nervous system. There's NMDA and AMPA. Now normally, only the NMDA receptor is on the surface of the postsynaptic cell, binding with glucose to continue the action potential. So if it's only NMDA, then there's no LTP. But when there's continued stimulation of the synapse by learning something, the postsynaptic cell begins to produce the AMPA receptor and put that in the membrane next to the NMDA receptor. And the presence of both of them strengthens the synapse. So if you have only NMDA, it's not LTP. If you have NMDA plus AMPA, that means LTP or basically memory. This effect can often be evoked using what's called associative learning. When nearby activity in the brain strengthens the synapse, causes the production and upregulation of AMPA receptors, then this synapse gets potentiated, this long-term potentiated. Associative learning is a very, very basic kind of learning. Almost anything can do associative learning, from an insect all the way up to a human being. It's usually just associating two things, usually two things that are close to each other in time. So, for example, with a lot of early neural systems work was done on Aplysia, which is a sea slug, very, very simple organism. It has a gill that it uses to get oxygen from the water. If you touch the gill is very fragile. The slug is going to withdraw it underneath a harder cover to protect it. So what researchers did was every time they touched the gill, they also touched the snail on the head. Over time, the snail associated that head touch with needing to withdraw the gill. And so eventually, the snail associated just the head touch with withdrawing the gill. So you could just touch the head, and it would pull the gill in. Right? So that's associative learning. The two stimuli are together in time, and the organism learns to associate one with the other. So the activity caused by poking the head and the activity caused by withdrawing the gills strengthened these synapses until all it took was a poke to the head to withdraw the gills, even though that's not a normal reflex for a sea slug. It doesn't do anything. Maybe a better known example, right, are Pavlov's dogs. What did Pavlov do to his dogs? Remember the story? Right?
Ringing a bell before the food is brought in to trigger the salivation glands.
Right, so in this case, the dogs associate the presence of food with the bell, and so eventually when the food was removed and the bell was sounded, the dog still salivated because they still reacted to the food. That was an association that they were given. Now, dogs have a much more complex brain than sea slugs, so it's a little bit more complex than that. But at its base, at its most fundamental, this works the same way that for you that it works for sea slugs. You can associate two things in time, and eventually that association is built so strong in the brain that it's really difficult to separate it, right? I'm sure you guys have some associations in your brain, whether it's a song that has a strong association with an event that you had, or an ex that you hate, and yet you cannot hear that song without bringing up that person's face in your brain. That's an associative memory. That's formed by LTP. I have, I don't know if I talked about this. Did I talk about my grandma's hallway? Yeah, I did, okay. So I won't repeat my stories. But yeah, smells are very good at evoking associative learning as well. So it's very strong smell stimulus can evoke an associative learning. There's also an opposite to this. So long-term potentiation increases the strength of synapses. There's also a phenomenon called long-term inhibition, or LTI, and that causes a long-term weakening of synapses. Right, but both of these processes contribute to learning and memory. Make connections, you break connections. You strengthen some synapses, you weaken others. That's all about learning and memory. All right, so LTP, LTI. Now, we know a lot about how brain neurons work, and we know a little bit about how things happen when we remember things. We also learn quite a bit about what happens when these processes go wrong, because they often produce symptoms, physiological symptoms, especially in humans, that need to be researched and treated. So, for example, we talk about neurotransmitters, right? What do neurotransmitters do? Where do you find them? Where do you find neurotransmitters?
The synaptic gap.
In the synapse, right? And what do? They carry information from one neuron to another in a chemical form, right? So they bridge the gap where one neuron uses an electrical potential, the other neuron uses an electrical potential, but in order to cross the gap, right, electricity doesn't cross a gap. So it has to be turned into a neurotransmitter, which crosses the gap, and then continues the action potential in the postsynaptic cell. And so if you can affect neurotransmitters in the synaptic gap, you can have a large effect on brain function. So schizophrenia is a neural disorder that's been associated with the neurotransmitter dopamine. There's evidence that increase in dopamine produced by amphetamines. Remember I talked about drugs of abuse, the rewards pathway. The reward pathway is mediated by the neurotransmitter dopamine, so drugs that you take that increase your dopamine makes you feel better, and that's why people take these drugs. Amphetamines increase your dopamine levels in your brain, and there's also studies that show that amphetamine use, especially long-term, produces the same symptoms as schizophrenia, which suggests that schizophrenia is also related to increase in dopamine levels in the brain. But we know that that's not the whole story, because we know that there is a genetic component to schizophrenia. And how we know that is by looking at the likelihood that two people will develop schizophrenia depending on their relationship. So if you are two unrelated individuals, you have a very low chance of developing schizophrenia. But if you have an identical twin and they are schizophrenic, you have up to a 50% chance of also developing schizophrenia. Just a regular sibling, you still have a higher than 0 chance of developing schizophrenia. grandchild, grandparent, same thing. So the further removed you are in terms of your relationship, the lower the probability is, which is a classic sign that genes are involved. So we know that it has something to do with dopamine levels, but we also know it has something to do with genetics. I already mentioned often in depression or bipolar disease, the neurotransmitter serotonin has been implicated. And the more modern drugs called SSRIs, selective serotonin reuptake inhibitors, have been shown to be effective in treating depression. So by allowing serotonin to remain in the synapse longer without being cleared out, that seems to improve depressive symptoms in people who suffer from those disorders. But as I said before, we're not really sure how that works. We know what SSRIs do. We know that people who take them feel better. But as far as what that does in the brain, still very much a black box. So addiction is a nervous system disorder, and here again is the reward pathway. Maybe this is a little bit more thorough than just me kind of going off on a tangent about it. So your reward pathway is composed of neurons that are called VTA neurons. VTA neurons are the ones that release dopamine when you do a good thing. And that's evolutionarily speaking, right? This is an evolved pathway. It's evolved in our brain to keep us doing things that perpetuate our species, right? That is, eat, sleep, and reproduce. Right? Those are all basically enjoyable things that we do. That activates the reward pathway in our brain, and so we feel good doing them, so we do them again. Now, drugs of addiction basically hijack this to make us feel good even when we're not doing those things. Some drugs like opium and heroin, they actually don't act directly on the reward system. What they do is there are inhibitors of the reward system. So your reward system can turn on, but it should also turn on, right? You're not going to be permanently in the state of in a high because you just had breakfast. So the reward system gets shut down after you're finished doing what the thing it is that you should be doing, right? That evolution says you should be doing. So you're not always high. But opium and heroin block that inhibition. So basically, you just keep that dopamine high going all the time. Okay? And that dopamine high is what makes us feel good. So of course we chase after it. That's what addiction really is at its base, is just keeping that reward pathway activated when otherwise it would be shut down. So cocaine and amphetamines have a different, attack a different part of the reward pathway. So instead of blocking the inhibition of the pathway, they block the removal of dopamine from the synapse. So these act very similar to serotonin reuptake inhibitors, which are used in depression, except these are dopamine reuptake inhibitors. And again, leaving dopamine in the synaptic cleft longer, we need to get a better feeling from the reward pathway, more stimulus, more output. So this is really, evolutionarily, this was to help keep us doing things which would perpetuate our species. But humans are really good at getting around things like that. So we realize that we don't feel good 24/7, and if you can't eat or sleep or have sex 24/7, how else are you going to stimulate the reward pathway to feel good? Well, lo and behold, a lot of these substances, which became drugs of addiction, got to be that way because they affect.
I'm not addicted to weed.
And that's why addiction is so difficult to overcome. Because really, right, this pathway is evolved in our brain, so it's very fundamental to who we are. So it can be very difficult to kind of try to disregard that or pretend it's not there. So yeah, so addictions can be a very difficult problem to treat because it's so deeply rooted in who we are as human beings. So neurodegeneration is another nervous system disorder. In this case, it's not synapses, but neurons themselves. Neurons are killed by an accumulation of abnormal proteins. These are called neurofibrillary plaques or tangles. This is a picture of a brain neuron these are the neurofibrillary tangles that surround it. They are proteins that are normally present in the cell and normally have a function. They do stuff. But for some reason that we still don't quite understand, sometimes they start to get in these clumps. And when they get in these clumps, not only do they not work, but they gum up everything else. And so eventually, these plaques fill the whole cell, and the cell can't function anymore. It can't produce energy, it can't fire action potentials, and then eventually dies. A number of neurodegenerative diseases have been diagnosed. Probably you're most familiar with Alzheimer's disease. Alzheimer's disease is characterized by generalized cell death. So cells all over the brain die from these neurofibrillary tangles. It's estimated that often Alzheimer's is usually a disease associated with old age, although there is an early onset version of Alzheimer's, but it's thankfully quite rare. But in adults who suffer Alzheimer's, it's been estimated that about 35% of their brain neurons have been destroyed by the age of 85. And of course, that affects things like memory, motion, all sorts of things like that. And eventually, of course, it kills enough of the brain cells that the basic functions like heartbeat and respiration don't work. It's fatal. Parkinson's disease is a little bit different. Parkinson's disease is neurodegenerative, but it focuses on dopaminergic neurons. Dopaminergic neurons, aside from being part of the reward pathway, are also part of the motor cortex and the motor pathways. And so that's often why people who develop Parkinson's often start with a tremor, because the dopaminergic neurons that are supposed to regulate your muscle activity die out. So if your muscles are always active, you would have this tremor, but we don't, because they're actually neurons that shut down the muscle when we don't want it to move. It says, don't move, stay quiet. But when those die, this random muscle movement just picks up and there's nothing to stop it, which is how you get these tremors. It's been estimated that 5% or more of the dopaminergic neurons in a Parkinson's patient die by the age of 85. There's a very interesting story around Parkinson's disease, which has to do with a drug called L-Dopa. So L-Dopa is the immediate chemical precursor to dopamine. It takes one very simple enzymatic step to turn L-Dopa into dopamine.
Just like taking teas.
I don't know if any of you guys are familiar with a movie called Awakenings. It had Robin Williams in it as a neurologist named Dr. Oliver Sacks. Anyone familiar with that movie? Or Oliver Sacks in general? So Oliver Sacks was a very well-known neurologist. He wrote a lot of books, including some sort of general knowledge books about the nervous system that are really interesting. My favorite is the one called The Man Who Mistook His Wife for a Hat. Although it's often very, very sad too when you hear about these people who have these very specific mental deficits from a very specific source. So early in his career, Dr. Sacks dealt with a number of people who were, it was a result of a disease, like fever or something like that. But they had become completely insensitive to anything. They had no reaction to anything. They lay in bed, you could do whatever, you could poke them, you could prod them, you could speak to them, they wouldn't answer. They would chew if you put food in their mouths, they would breathe, but otherwise they were completely non-reactive after they had this fever. And so obviously the families and the relatives of these people were desperate because these were generally healthy younger people. These weren't Parkinson's patients. Healthy younger people who had somehow just stopped working. Sachs thought it might be partly the dopaminergic neurons in the brain that were selectively destroyed by this disease, whatever it was. And so what he did was he fed these patients L-DOPA intravenously. The idea being that even if you have very few dopaminergic neurons, if they get this L-DOPA, they can very easily make dopamine. And so even a few neurons that are still around can easily make dopamine, and if dopamine is present, then the brain will start acting normally again. And what happened was he fed these patients L-DOPA, and they woke up. That's how the movie's called Awakenings. They woke up. They were completely normal. They had no idea what had happened to them. In some cases, they had aged years, decades. Family members had died, but they had no recollection of this. They just woke up. But, you know, it was obviously to the families of these people very heartening that they were now alive and well and recognized them and could talk and whatever. So it was really a tremendous advance. Unfortunately, because these people had lost so many of their dopaminergic neurons, that even with L-DOPA, over time, their dopaminergic neurons began to die out. And so even giving these people L-DOPA, they slowly went back to this inactive sleeping state. They were only active for a few weeks to a few months, and they started to fall back into the state, and they couldn't be reawakened from that. So it didn't end up all that happy an ending, but at least they got a little bit out of it. There's actually another story from San Francisco in the 60s, the story of the frozen addicts. So there was a case where somebody had made, I think it was the drug MDMA, but they made a mistake when they did the chemical synthesis. It became something, a slightly different drug. I'm not sure exactly how, but a number of people who took that particular dose of that particular drug kind of got like the Awakenings people. They got completely frozen. They couldn't move. And we know that dopamine is involved in pathways that regulate movement. And so people thought the same thing, which was, well, if the Awakenings people got L-DOPA and woke up, maybe we can try it with the frozen addicts. And they did, and the same thing happened. The frozen addicts woke up, they were able to move, they were able to talk, communicate. But again, over time, the L-DOPA was losing its effect. And so eventually they fell back into that frozen state. But whether it's a natural cause or a disease or a drug, the loss of dopaminergic neurons can be catastrophic to people. So I think I've mentioned this before about adult neural stem cells. So all these diseases are really depressing, right? I don't like to depress you all like, oh great, this is what I have to look forward to. But it's actually not all bleak. So the dogma used to be that you never made any new brain cells. You were born with all the brain cells you would ever have, and all you do is kill them by going out drinking beer on Saturday nights as a college student.
Well, more than just.
But in fact, no. But in fact, the dogma is wrong. In fact, the brain is always producing neurons, even into old age. These are called adult neural stem cells. So these guys here, they are by themselves generalized regular old cells, but when they divide, one of their offspring can become a neuron. And stem cells are all over the place. Stem cells are all over the body. There are stem cells that divide and produce immune cells. There are stem cells that divide and produce red blood cells. And there are stem cells that divide and produce neurons. We did not know of the existence of adult neural stem cells for a long time. And the reason that these were discovered, and they weren't discovered until the 1980s because of AIDS. So in the 1980s, right, AIDS was, HIV and AIDS were a major issue. One of the earliest treatments to treat this viral disease was a drug called BRDU. So it controls and shuts down the replication of the HIV virus. so it doesn't replicate so much in your body. But BRDUs also has an interesting property that it's incorporated into cells that are undergoing mitosis, and only cells that undergo mitosis. So people who had AIDS who took the drug, all of the cells in their body that underwent mitosis, these were adults, right? These were people in their 30s, 40s, 50s, were labeled with this BRDU, which could be detected. And so when they died, oftentimes they donated their bodies to science, and the scientists looked in the body for where are the cells that incorporated BRDU, because those would be stem cells, dividing cells. And lo and behold, a bunch of cells in the brain lit up. And these were cells nobody had ever known about. Nobody knew that there were adult stem cells in the brain. And so it was a real awakening, no pun intended, right? Because yeah, the dogma used to be, no, you have no new brain cells. You're screwed, right? Now we know that's not true. There are adult neural stem cells. The brain can regenerate neurons. It's not perfect, but it does provide hope for people who do suffer from neurological diseases that we might be able to someday come up with drugs which will tell these neural stem cells, make more dopaminergic neurons. We're running out of them. So we'll see, but it's not all bleak. Okay, so a couple things. First of all, there's a quiz this weekend. It's going to cover Wednesday's lecture on nervous system organization and today's lecture on learning and memory. Okay, so it's going to open right after class. It's going to close right before class on Monday. The other thing you guys have to do this week is your abstract for your poster on learning. So I did put up the
I've been getting emails from people.
the groupings
I got you
on
and I didn't
Google,
respond to it.
but if you haven't looked at them yet, I printed them out for you. So find yourself in your group. And I'm afraid I had to use the names that you have on Moodle, so it may not be your preferred name. I apologize for that. But what you need to do this weekend with your group is get together either virtually or in person and talk about the topic
Virtually.
of your poster on learning.
Coming.
And remember, we talked about a couple of different alternatives for a topic. You can talk about learning strategies or physical strategies for enhancing learning, right? Like exercise.
I know.
And you can look at different forms of exercise. You can look at aerobic exercise, anaerobic exercise. You can look at particular exercise regimens that might be that might help cognition.
Yeah, she says.
You can look at mental or emotional ways of improving cognition, things like meditation, cognitive behavioral therapy, CBT. You can look at drugs, chemical ways to improve your cognition, or you can look at ways to overcome learning disabilities. Drugs like Ritalin, which help people who have ADHD be able to focus better.
My simple pleasure watching that Espanol.
So, what I'm going to do is everybody who's listed first in their group, so everybody who's listed first, I'm going to make you kind of the group facilitator. So, try to, you can reach out, email everybody in the group, try to connect with them. That doesn't mean that if you're not top of the list, that doesn't mean you don't have to do anything. All right, when the facilitator reaches out to you, reply, right? Keep the lines of communication open. So, what I want you to do this weekend is work on this poster abstract. And the abstract is going to ask you to give a title for your poster based on what type of cognition or learning improvement you're going to focus on. Keywords, right? The theme, which is going to be physical, mental, emotional, chemical learning disabilities. And then a short abstract, which kind of gives the gist of the poster. It doesn't have to be perfect. It doesn't have to include everything, because a lot of times when you're making the poster, things change. So the abstract is due earlier than the poster. It doesn't have to be a perfectly faithful representation of everything your poster is going to talk about, just generally. This is something that's always gonna be true in science. Every time you go to a scientific conference, the abstract is often due months before the actual conference where you're presenting a poster. So a lot of the times when I make a conference abstract, I have no idea what the poster's gonna look like. I haven't done the experiment yet. So I kind of try to make this like nice, mushy kind of BS thing that would cover almost any eventuality, and then I actually do the experiments, and 3 or 4 months later, when I actually make the poster, then I know what it's gonna be done. And I hope the abstract is close enough. So a lot of times the abstract is not perfect. That's okay. That's part of writing posters. So there's a Dropbox on Moodle to submit your proposal, which is due Monday by midnight. It's a group submission, so only one person needs to submit. So I'm going to make it the,
Let's go.
I think the last person on the list. First person on the list is the facilitator. The last person on the list is responsible for submitting your proposal to Moodle. Got that?
That's the.
Ooh. This one.
So, yes.
So, remember you got the form, and the form is also electronically on Moodle. It's part of that assignment. Click on the assignment, you'll see the form. Just fill out the second page and upload that. You don't have to upload the first page with this stuff on it. Just the last page.
I know, but I don't know.
Please, when you
Is it 20?
save the proposal file, save it with this type of file name. So, last name of everybody in the group, alphabetically, underscore your poster title. If you save it like that, then it'll be really easy for me to figure out whose abstract goes with whose poster. Question?
So I guess, so we're taking one of the themes, either physical, mental, chemical, or like strategies to overcome. So we're basically picking one, and then are we doing any like research and like finding stuff about it, basically, right?
Right, I mean, how are you gonna write a poster about it if you don't know anything about it, right? So, yeah, so you're gonna choose a topic, but then you're gonna start researching it, right? You can use the biomedical literature, you can go through the
Just
library
Wikipedia.
website, there's lots of really good indices and
Where's
databases that you can search through.
Sure.
It doesn't have to be exhaustive, but if you have maybe a half dozen good sources, that'll help you put your poster
Okay,
together.
hands got born.
So do
Talk
we
more
need to
about
have that with
Hands be
the
born.
abstract part, or is
All
that
these
the?
dating apps.
That's where the poster part, so yeah, that's why I mean, like, the abstract comes before you do a lot of the work, so sometimes the poster is different when you get to the end, that's
Yeah, I
okay.
don't know. I
So the
think it should
abstract
be
is
on
like
here
the
just to be
overview of like
sure.
what we wanna talk about.
I just pay attention at that point.
So it's basically the idea, you've all